Hi, welcome to another movie for our molecular cell biology class. In this movie, we are going to be addressing the question of how stable is the DNA, and specifically, we are going to be talking about the mechanisms that are used to repair that DNA if it is ever damaged. So the first essential idea is that DNA is continuously being damaged, and that damage is due to its own chemical nature. So there are many different things that affect the structure of the DNA continuously all the time. One of the things that happens to our DNA all the time is deamination. And the main effect, the main target of deamination is cytosine residues, which when they become deaminated, they originate uracil residues. So that's a C to U mutation that occurs naturally in our DNA. And like that, there are many other things that affect our DNA. The other type that is important to keep in mind are depurination and depyrimidation events. So the losses of purines and pyrimidines, which happen all the time in our DNA. And whenever that happens, they trigger the appearance of what is known as an AP site. AP is standing for apurinic or apyrimidinic site in our DNA. So all of those are things that happen normally to our DNA because of its own chemical nature. So the question is, whenever a change happens in our DNA, is there a way to repair that type of DNA damage? And the answer is yes. And there are 4 different mechanisms that can be used to repair DNA damage. And those 4 mechanisms are direct reversal, excision repair, translation DNA synthesis, and recombinational repair. All right, so let's start with direct reversal of DNA damage. So direct reversal is the direct elimination of whatever change has taken place in the DNA. So the best example of this kind of direct elimination of the damage the DNA has suffered is the direct repair of thymine dimers by a process known as photoreactivation. So we had already discussed in class that exposure to UV light can actually trigger the formation of these structures known as cyclobutane rings, also known as thymine dimers. In fact, they can actually happen whenever you have 2 pyrimidines, one right next to the other in one of the strands of DNA. So these cyclobutane rings can happen whenever you have 2 thymines, or whenever you have a thymine right next to a cytosine, or whenever you have 2 cytosines right next to each other. Of course, the order can be altered, and still that will allow the formation of thymine dimers. So whenever you have any of these 4 different combinations in your sequence of DNA, exposure to ultraviolet light can actually trigger the formation of those cyclobutane rings, also known as, when it actually happens with adjacent thymines, they are also known as thymine dimers. All right, so those structures are bad for DNA because you can easily imagine how any type of enzyme that is using DNA as a template to either produce another copy of DNA, such as DNA polymerase, or to produce an RNA molecule, such as an RNA polymerase, will have a very difficult time going through that particular section of the DNA. The enzyme will know how to read those 2 nucleotides that had been linked by those cyclobutane rings. So that will block the elongation step of either DNA replication or RNA synthesis. Therefore, there is a need to repair that kind of damage in the DNA. And the easiest way, the most direct way to do it, is by the engagement of this enzyme, an enzyme known as DNA photolyase. And this enzyme has the ability to simply break those cyclobutane rings directly. This enzyme is activated by UV light, and therefore the same process that generates the formation of those cyclobutane rings is the same process that activates the enzyme that has the ability to eliminate the damage. As you can see, there is no need to resynthesize anything at all. The enzyme by itself has the ability to hydrolyze those cyclobutane rings, therefore producing the normal structure of the DNA, eliminating the damage in a direct way. Now, this is a very effective, very rapid mechanism to revert that kind of DNA damage, and it actually represents the most efficient way to eliminate that kind of mutations in your DNA, those cyclobutane rings. However, the issue with this is that this particular mechanism of DNA repair is not present in humans. And this is kind of an interesting fact. There are many organisms that do contain this enzyme, do encode this particular enzyme, but that particular enzyme is absent in all mammals. So our common ancestor for all mammalians lack that particular enzyme. It got lost. And one funny piece of information is that is partially a reason why it is believed that the very early mammals were nocturnal animals. In the absence of activity during daylight, it's really easy to see how there was no selective pressure to maintain the gene coding for this particular enzyme. But it would be great if we could put it back in place, because then the frequency of skin cancer in humans could be dramatically decreased if we had this mechanism of direct DNA repair. All right, let's move on with the second mechanism that we know of, direct reversal. And the second example of direct reversal involves the elimination of methyl residues that can occasionally become attached to guanine residues, therefore producing methyl guanine residues. So methylation of guanine alters the chemical nature of guanine residues, and therefore that methyl group needs to be eliminated. There is one very specific enzyme known as methyl guanine methyltransferase that has the ability to specifically recognize those guanine residues that have been methylated, and this enzyme not only recognizes that event but also interacts with that specific guanine residue and eliminates the methyl group, and then passes that methyl group to another receptor. So it cuts off the methionine of the guanine residue and goes on and conjugates that methyl group to another target. So it methylates another target, therefore eliminating the methyl group from the guanine residue that had been methylated. Now, there is one thing to keep in mind related to this, and that is that opposite to the previous mechanism of direct reversal that we talked about, the activity of the photolyase, the enzyme that is involved in the direct elimination of methylation events from guanine residues is present in humans. So this enzyme exists in humans, and therefore this direct reversal is actually present in humans. Another important thing to keep in mind when it comes down to this methylation event, guanine residues are not the only ones that get methylated. Cytosine residues are also methylated. But methylation of cytosine residues to form methyl cytosine residues is actually something that is not considered to be a mutation. Instead, methylation of cytosine residues is very intentional. There is a very specific mechanism to mediate the methylation of cytosine residues, and methylation of cytosine residues typically occurs in sequences known as CPG sequences, meaning cytosines that are right upstream from guanine residues are the ones that can be methylated. And one final idea related to methylation of cytosine residues, DNA methylation. When you methylate cytosine residues in DNA, typically has the effect of silencing that specific region of the DNA. And the silencing is actually mediated by the formation of heterochromatin. And one final thing, this is the mechanism that is used to silence lines and signs. So lines and signs are silenced, that is, they are kept from becoming active, from becoming transcriptionally active by methylation events that involve cytosine residues. All right, so that's all about direct reversal. So let's move on to the second mechanism of DNA repair, and that is excision repair. And excision repair is all about creating a small gap in one of the two strands of DNA so that the other strand of DNA can actually be used as a template to synthesize the complementary strand. So excision repair is all about excising a section of the DNA. And that excision event can actually involve either the elimination of a single base, and in that case we refer to it as base excision repair, or it may actually involve the elimination of a larger section of sequence, and that is nucleotide excision repair. And there is another mechanism of excision repair that is actually the one that happens while DNA is being synthesized. So it's one that is directly responsible for further decreasing the frequency of mutations during DNA replication, and that is mismatch repair. So we are going to talk about all three different mechanisms of excision repair. So the first one, base excision repair. In this specific mechanism, all you're going to be excising is a single base. So even though you excise only one base, in the end you end up with a gap of only one nucleotide. So in the end, you end up having to fill in the gap one way or the other. And that will always require the same machinery that is used during DNA replication of the lagging strand. So keep that in mind. Whenever you're creating a gap in the DNA sequence, filling in the gap that you're creating will require the same DNA replication machinery that is used during lagging strand DNA synthesis. All right, so let's go through the mechanism of base excision repair. So the first thing that is always required to repair DNA damage is to identify the place where DNA damage has taken place. So when it comes down to base excision repair, that mechanism typically corrects the presence of uracils in the DNA. So there is a specific enzyme that is capable of recognizing whenever you have that kind of mismatch in the DNA, and that enzyme brings a DNA glycosylase, which has the ability to break the link between the sugar and the base. So the DNA glycosylase breaks the link between the sugar and the base, therefore getting rid of the base. If you break the link between the sugar and the base will be gone. And whenever the base is eliminated, then you end up with an AP site. And as I had indicated before, an AP site stands for an apurinic or apyrimidinic site. Now, once you have eliminated the base, then you need to break the backbone of the DNA. And that breakage requires an endonuclease activity. And it's an endonuclease activity that will actually take place right where you have the AP site, the apurinic or apyrimidinic site. Therefore, the enzyme that will be doing that is known as an AP endonuclease. AP endonuclease, AP because it's recognizing an AP site, and endonuclease because it is breaking within DNA, within an intact DNA backbone. So it's creating this gap now, this small breakage, this small nick. This is what we refer to as a nick. And that nick in the phosphate backbone of the DNA then helps recruit another enzymatic activity. In this particular case, one that will be breaking the phosphodiester bond between that particular sugar and the next nucleotide. So since it is a phosphodiesterase, that's the activity, breaking a phosphodiester bond requires a phosphodiesterase. And the link that is being broken is between the sugar, which is deoxyribose, and the next nucleotide. The enzyme that will be doing this job is known as a deoxyribose phosphodiesterase. So that's the next enzymatic activity that is required to perform base excision repair. So the first component is DNA glycosylase, the second one is an AP endonuclease, and now the third enzymatic activity is a deoxyribose phosphodiesterase. Now, once that enzyme has eliminated altogether the sugar and the phosphate group that was in that location, now you have a real gap in the DNA sequence, and in that particular gap you have a 3' end and you have a 5' end. So that 3' hydroxyl group that is located right there can actually be used as if it was a primer to add the next nucleotide, and that is the job of DNA polymerase, specifically the one that is typically engaged in ligand strand synthesis, and that is DNA polymerase delta. Once DNA polymerase delta puts in the nucleotide that had been eliminated, now the next thing that needs to be done, and I marked the wrong nucleotide right there. This is the nucleotide that was added. Now you have you have a nick in the backbone of your DNA, because now you have a 3' hydroxyl and you have a 5' in which you only have one phosphate. So you need to now link these two, and that is the job of the ligase. So you need a DNA ligase to seal that nick that was left in place, and that's the last enzymatic activity that is required to perform base excision repair. Now, I hope that by looking at the complexity of this process, we have 1, 2, 3, 4, and 5 different enzymatic activities that are required to perform base excision repair. I hope that it is pretty obvious to all of you why this is substantially less efficient than direct reversal of DNA damage. And this is one of the excision repair mechanisms. This is base excision repair. All right, let's move on now with the next one. Keep in mind the type of errors, the kind of DNA damage that can be repaired by base excision repair. It typically will be the mechanism that will be used to fix a deamination event. So a deamination event in which you have a cytosine, and the deamination of cytosine triggers the formation of a uracil residue. That kind of event typically is repaired by base excision repair. All right, let's now move on with nucleotide excision repair. Let's go on to the next one. Now, nucleotide excision repair allows for the cell to fix larger types of damages that may actually take place in the DNA. For instance, the addition of a bulky chemical group to a base in the DNA. The only way to eliminate those bulky modifiers that are attached to some of these bases is by nucleotide excision repair. Now, the other thing that nucleotide excision repair is very efficient at eliminating is those cyclobutane rings. Cyclobutane rings that are produced by UV exposure. Now, keep in mind, the most efficient way to eliminate that kind of cyclobutane rings will be direct reversal. But the thing is that in humans, we don't have that mechanism. Therefore, the way by which we eliminate that kind of mutations whenever they occur in our DNA is by nucleotide excision repair. There are other types of DNA damage that can also be eliminated by nucleotide excision repair, and in general, it is important for you to think of nucleotide excision repair as the most multitask type of DNA repair that we have in our body. Is the type of DNA repair that can deal with a large variety of things that may actually affect our DNA. All right. Now, when it comes down to nucleotide excision repair, you can imagine that the type of damage that will be repaired by nucleotide excision repair can happen in two different locations within our genome. One of them is it may actually fix damage that happens anywhere in our genome, but specifically in regions that are not being transcribed. So when it is used to fix damage in regions that are not being transcribed, it is referred to as global genome nucleotide excision repair. So this is when it fixes damage in regions that are not transcriptionally active. So global genome nucleotide excision repair is used to repair DNA damage in regions that are not transcriptionally active. And what I have tried to show you here is the way that this could actually fix the type of damage that could be produced by UV radiation. So this is your arm. That thing right there is your arm. Not the best picture, but that's the idea. These are your fingers. All right, so you have your arms, and those are typical places in our body where we typically don't protect them too much. So you have a short sleeve shirt, and you're walking somewhere in the UTEP campus because coronavirus is not a thing anymore, and you get exposed to sunlight, and that sunlight hits your arm, and in your arm, on the very surface, you have epithelial cells, and in that epithelial cell, you have your nucleus, and your nucleus is your DNA, and that DNA is being damaged by UV light. And that UV light is triggering the formation of pyrimidine dimers. And those pyrimidine dimers need to be fixed, but you can easily imagine that they could be happening either in transcriptionally active regions of your DNA or in DNA that is tightly packed and transcriptionally inactive. So depending upon where the damage is happening, you will invoke different mechanisms, different repair mechanisms. So if it happens in the transcriptionally inactive DNA, then you will invoke global genome nucleotide excision repair. Whereas if it were to actually happen in transcriptionally active regions of your DNA, regions that are being actively transcribed into RNA, then the mechanism to repair that type of damage will be transcription coupled nucleotide excision repair. All right, so let's go through both of those different variations of nucleotide excision repair. Now, before doing that, keep in mind one thing. The only difference between these two different mechanisms of nucleotide excision repair, global genome versus transcription coupled nucleotide excision repair, is who detects the DNA damage. The enzyme that does the very first thing, and that is finding the DNA damage that needs to be fixed, that enzyme is the one that is different between these two different mechanisms. Other than that, all other enzymes are shared by both mechanisms. So again, let me just re-emphasize this. The only difference between these two mechanisms of DNA repair is who finds the damage. All other steps in these repair pathways are absolutely identical. So let's start with global genome nucleotide excision repair. So the idea here is relatively simple. The first thing that needs to happen is there needs to be enzymatic activity that will identify the region of DNA where the damage is located. Next, it actually needs to recruit a series of enzymes that will do a couple of things. Number one, it needs to break the double-stranded nature of that particular section of DNA. So it needs to open up the DNA. So it needs to open it, open the DNA, and that will be a helicase activity in order to open the double-stranded DNA molecule. And the other thing that is required is you also need to recruit enzymes that will cleave around the region that contains the damage. So you need enzymes that will cut within the DNA, and enzymes that cut within the DNA are referred to as endonucleases. So you need to recruit endonucleases that will cut around the region that contains the DNA damage, so that now, since you have already opened the double-stranded molecule and now you cut on the backbone, now the section of DNA that contains the damage can be eliminated. So DNA damage is eliminated. And now what you end up with is a gap that needs to be filled in. And that filling in will be the job of DNA polymerase. And once you have filled it in and now you just have a nick in your DNA structure, the nick is sealed by the activity of ligase. So those are the last two steps that will be required in order to fix the DNA damage that has been fixed by nucleotide excision repair. All right, so let's now go into the specifics of this mechanism of DNA repair. So what are the enzymes? What are the specific identities of the enzymes that are engaged in this pathway? Well, so the identities of those enzymes, of many of the enzymes that participate in this particular pathway, were identified thanks to the presence of a particular type of disease in humans. A disease that is associated with very high sensitivity to UV light. And in fact, this specific disease is manifested by the presence of really extreme damage in any region of the body that is exposed to UV light. And the name of the disease is xeroderma pigmentosum, or XP. And individuals that are affected by this particular disease have extreme sensitivity to UV rays from sunlight. And the main manifestation of this very high sensitivity is the development of extensive photo damage in any region of the body that has been exposed to UV light, and that leads to the formation of cutaneous malignancies, severe ophthalmological abnormalities. So people have such extreme sensitivity in their exposed regions, not only of the skin but also the eyes, that they end up typically losing vision early on. And these individuals also end up dying at an early age due to skin cancer. So all of these are symptoms typically associated with xeroderma pigmentosum. Now, the characterization of these disease, the identification of individuals with this type of disease as being due to exposure to UV light, led people to think that if they could actually use cells derived from patients that exhibit this disease, they could probably characterize the DNA repair mechanisms that are involved in fixing the type of damage that is associated with exposure to UV light. And so following that logic, scientists were able to characterize the different enzymes that are involved in that particular pathway, in the pathway of global genome nucleotide excision repair. And because of that association with this disease, many of the enzymes that were identified were then denominated XPA or XPB or XP whatever. So many of the enzymes, the names of many of the enzymes involved in this pathway starts with XP. All right, so now one important point that needs to be made. Even though this disease is typically associated with skin cancers, when parents notice that their kids exhibit that very high sensitivity to exposure to UV light, they can actually establish measures early on that will prevent exposure to any type of UV light. And if that is actually done early on, the individuals that are affected by this type of defects in this DNA repair mechanism can actually have fairly normal lifespans and fairly normal phenotypes, as shown by these two individuals. These two young kids, they have been protected their entire lives from any type of exposure to UV light, and as you can clearly see here in these two pictures, their skin looks absolutely normal. And that's simply because the main type of DNA damage that is repaired by nucleotide excision repair is cyclobutane rings, the type of damage that is associated to exposure to UV light. All right, so moving on. So now let's go into the enzymatic details of this repair mechanism. So the first stage involves the identification of the DNA damage. So the very first thing that needs to happen is there needs to be an enzyme, a protein that will recognize the kind of distortion that is triggered in the DNA by the presence of those cyclobutane rings. And that is the job of Scleroderma pigmentosum C. So think of C as the seeker, the protein that seeks for the damage, the one that is searching through the DNA for that kind of damage. So C, seeker. Scleroderma pigmentosum C is the seeker, the one that seeks for the damage. And whenever it finds that kind of damage in the DNA, then it binds to that particular region in the DNA, and then it recruits the next 3 players in fixing this type of damage, and they are Scleroderma pigmentosum A, and Scleroderma pigmentosum A actually is a factor that helps recruit the next factor, and that is a transcription factor. So these two come together, XPA and transcription factor 2H, they come together. Now notice this is a transcription factor. Let's spell it out. TF2H is transcription factor 2H. Now, the name of that transcription factor says a lot about what it does. Transcription factor 2 means it is associated with RNA polymerase 2, which is the one that is involved in synthesizing messenger RNA. So this specific transcription factor is essential for the synthesis of messenger RNA. And what this transcription factor actually contributes to RNA synthesis is that it contains a helicase activity that allows for the opening up of the double-stranded DNA molecules, so that now you can produce single-stranded templates that can be used by the RNA polymerase to synthesize the RNA molecules that need to be produced. So in this case, this transcription factor is contributing the same kind of activities, contributing the helicase activity to the DNA repair mechanism. And the helicases are actually 2 different proteins. There are 2 proteins that are endowed with helicase activity, and those are Scleroderma pigmentosum B and Scleroderma pigmentosum D. So B and D are helicases, and they are subunits that are found within TF2H. So let's say that again. Scleroderma pigmentosum C is the first protein involved in nucleotide excision repair, is the one that seeks the damage, and once it finds the damage, it recruits Scleroderma pigmentosum A, which then helps recruit transcription factor 2H. And transcription factor 2H is a large protein complex that contains many different proteins, but among them it contains 2 other proteins that have helicase activity, and they are Scleroderma pigmentosum B and Scleroderma pigmentosum D, which are the ones that are going to open up the double-stranded molecule of DNA, as it is represented in the next part of this diagram. So now you have TF2H engaged in there, and you have Scleroderma pigmentosum B and D. and they are the helicases. Now, the moment that you open up the double-stranded DNA molecule, you generate single-stranded segments of DNA that need to be stabilized. They need to be prevented from forming a double-stranded structure again. And that is the job of replication protein A, which we had already talked about it when we talk about DNA replication. So replication protein A is a single-stranded DNA binding protein. And the whole job of this protein is to keep the two strands of DNA from being able to form hydrogen bonds with each other again, therefore reforming the double-stranded structure of the DNA. So RPA is our next player that is recruited into that specific region. And now what you have created is a bubble in the DNA, a bubble that is located right on top of where the DNA damage took place. So now what you need to do is you need to recruit the endonucleases. And the first one to be recruited is XPG. So XPG is recruited downstream from the place where the DNA damage took place. So this is this DNA structure. You can think of it as going 5' to 3'. So downstream, meaning to the 3' end of the damage, XPG is putting that 3' end of the region that contains the DNA damage. And now the next step will be to put the endonuclease on the other end, toward the 5' end of the damage. And that enzyme, that other endonuclease, is XPF, which comes together with another protein known as ERK1. So XPF doesn't come by itself, it actually comes associated with another protein known as ERK1. So those are the two endonucleases, XPF and XPG. Again, they are both endonucleases. And their job will be then to cut the DNA in those two regions. So the endonucleases, now XPG and XPF, will cut your DNA, therefore releasing the region that contains the DNA damage. And that creates a gap in your DNA, a gap that needs to be filled in. And remember, this is a 3' hydroxyl group. This is a single 5' end with a single phosphate. So now you have to fill that in, and that is the job of DNA polymerase. Specifically, that will be the job of DNA polymerase delta. And you also will need then a ligase to seal the nick that will be left in place. So after DNA polymerase does its job of filling in the gap, then you need ligase. And ligase will be the last enzymatic component required to fix that kind of damage. So that's the complete set of enzymes that will be required for nucleotide excision repair, specifically for global genome nucleotide excision repair, which will be fixing cyclobutane rings that are forming in DNA that is not being transcribed. All right, so that's global genome nucleotide excision repair. Now, for DNA that is being actively transcribed, the mechanism to fix the DNA damage will be transcription coupled nucleotide excision repair. And the important idea here is that since that DNA is transcriptionally active, whenever you have a cyclobutane ring in your DNA, what that cyclobutane ring in the DNA template is going to trigger is the RNA polymerase, which is transcribing your DNA, ending up stuck in place. So it is stuck, it can't move on because it doesn't know what to do with the cyclobutane ring, and therefore it is set to be halted. It has been stopped on its track. It cannot move any further because it has found those cyclobutane rings, and it's now waiting for the DNA damage to be fixed. And that is an essential idea because the detection mechanism now that will be engaged in fixing transcription coupled nucleotide excision repair is started by recognizing a halted RNA polymerase. That is the signal that is recognized by this repair mechanism. All right, so just like for global genome nucleotide excision repair, there was a human disease that allowed us to characterize the enzymes that are involved in that repair mechanism. For transcription coupled nucleotide excision repair, there is also a human disease that allows us to recognize the specific enzymes that were characteristic of this particular pathway. And that disease is known as Cockayne syndrome. And the name is not actually showing up here. Oh, there you go. Cockayne syndrome. So this is the name of the human condition that is associated with defects in transcription coupled nucleotide excision repair. Now, just like for Scleroderma pigmentosum, the enzymes were known as XP something. For Cockayne syndrome, the enzymes that are specific of this pathway were then called Cockayne syndrome whatever. And there are only two of them, Cockayne syndrome A and Cockayne syndrome B. Those are the two specific enzymatic players that are specific of this pathway. Now, Cockayne syndrome is a rare autosomal recessive disease in humans, so the frequency is very low in humans, but individuals that are affected by this syndrome, Cockayne syndrome, exhibit a very striking phenotype that can be summarized as defects in the development of the central nervous system. So they have photosensitivity, they have microcephaly, they exhibit slow growth, and they also exhibit premature aging. But the most striking features are all associated with the development of the central nervous system. So microcephaly being the most striking feature, but also there are developmental problems altogether associated with neurological development, which is interesting and indicates that transcription is quite essential and magnified when it comes down to the development of our brains early on during pregnancy, as well as very soon after birth. So transcription is just playing a critical role everywhere, but even more in terms of the development of the central nervous system. All right, so again, there are only two specific enzymes that are characteristic of this pathway, Cockayne syndrome A and Cockayne syndrome B, and they together are the seekers in this particular pathway, the ones that seek for the damage. So these two proteins all together, Cockayne syndrome A and Cockayne syndrome B, what they do is they recognize the halted RNA polymerase, because that's the signal that triggers this particular pathway. You have an RNA being synthesized by RNA polymerase, and whenever it finds those cyclobutane rings, the enzyme just simply doesn't know what to do. So it remains there in place. It was reading the DNA template and it was moving along the DNA template fairly quickly, but whenever it hits a region that contains those cyclobutane rings, the enzyme is now completely stopped on its tracks. It can't continue moving along, and now it's just there sitting in that particular region that contains the DNA damage. And that forms a signal that is recognized by these seekers, Cockayne syndrome A and Cockayne syndrome B. The moment that they recognize that damage, they interact with the DNA, and then they start the recruitment of all the other factors that we talked about before when we were talking about global genome nucleotide excision repair. So the first additional proteins that will be recruited will be Scleroderma pigmentosum A, transcription factor 2H, and replication protein A. So the idea is the only enzymes here that are missing is the seeker for Scleroderma pigmentosum. So the seeker, sorry, for global genome nucleotide excision repair, the seeker for global genome nucleotide excision repair is scleroderma pigmentosa C. Remember that. And that is the one that is absent in this particular pathway. So there is no XPC here, but all the other enzymatic players, XPA, RPA, transcription factor 2H, which contains the helicase, XPB and XPD, remember those are the helicases, all of the other players are recruited, and they are recruited by cocaine syndrome A and cocaine syndrome B. So from that moment on, the moment that those other players are recruited, the pathway just follows exactly the same dynamics that we have described for global genome nucleotide excision repair. Meaning, after you open the double-stranded molecule because of the helicase activity of scleroderma pigmentosum B and D, the next thing that needs to happen is RPA associates to a single-stranded DNA, keeping it in the single-stranded shape, and then you have the endonucleases being recruited, XPG and XPF, which are going to cut upstream and downstream from the region where the DNA damage is located, and then that allows for the dislodging of the segment of DNA that contains the damage, leaving behind a gap that needs to be filled in by the polymerase activity of DNA pol, and that activity then is going to leave a small nick in the backbone of the DNA that needs to be sealed by the activity of DNA ligase. So it's exactly the same mechanism that we had described previously for global genome nucleotide excision repair. All right, and that's all you need to know about transcription coupled nucleotide excision repair. Moving on, the next mechanism that basically engages nucleotide excision repair is what is known as mismatch repair. And the difference here is that this mechanism is associated with DNA replication. So this is happening while DNA is being synthesized. And one important idea that I really want you guys to remember is that DNA synthesis, DNA replication, is happening during the S phase. So this type of DNA repair is specific of S phase as well, because it happens only during DNA replication, which takes place during the S phase of the cell cycle. And just as a quick reminder, the cell cycle basically starts with mitosis. It's followed by the G1 stage, followed by then the S phase, which is where DNA replication takes place. This is then followed by another gap stage, gap 2, and that's followed then by mitosis. So when it comes down to the number of chromosomes that you have at every stage, you have 2n here in G1. During S phase, you go from 2n to 4n at the end of S phase, because that's when you're replicating your DNA. So you start with your diploid genome and you end up with twice the diploid genome because you have now replicated your entire genome. So at the end of S phase, you have a 4n chromosomal number, and you remain in 4n until mitosis. At the end of mitosis, you regain again your diploid status in the cell. All right, so that was just a reminder of what's happening during the cell cycle. So this repair mechanism, mismatch repair, is exclusively taking place during DNA replication. And the idea is that even though DNA polymerase has a very low error rate, and remember, the error rate for DNA polymerase is about 1 error every 1 × 10 to the 8 base pairs. So there will be about 64 errors per every round of DNA replication. Now, thanks to this specific mechanism of DNA repair, mismatch repair, the frequency is then broke down to basically nothing. Because whenever there is a misincorporation event that takes place during DNA replication, whenever the DNA polymerase makes a mistake, that mismatch that has been created by the mistake that was done by DNA polymerase is now fixed by mismatch repair. All right, so this mechanism has not been fully characterized in humans. In bacteria, it is actually mediated by 3 different enzymes known as MOT-S, MOT-H, and MOT-L. And the name MOT is simply associated to the fact that whenever you have mutations affecting those enzymes, the frequency of mutations in general during DNA replication goes substantially higher. It shows a dramatic increase in the frequency of mutations during DNA replication. So the enzymes that are involved in this pathway were denominated MOT. And there are 3 different enzymes: MOT-S, which is in this particular case the one that senses the damage; MOT-H, which is an endonuclease, creates a hole in the DNA; and then there is MOT-L, which enhances the activity of MOT-H. Now, in humans, what we know is that there are 2 homologs of the enzymes that are present in E. coli. So in E. coli, you have 3 different enzymes. In humans, there have been only 2 that have been identified, and they are homologs to specific components of the machinery found in E. coli. So because of that, because of being homologs, they are referred to as MOT-S homolog and MOT-L homolog. So notice that the one that is missing is MOT-H homolog. So there is no MHH. So there is no homolog to MOT-H in humans. So, okay, so the idea here is that one of those 2 enzymes, specifically MSH, is the one that finds the damage, and then it activates the endonuclease activity of MLH, and that endonuclease activity happens 5' from the place where the error was made. So then there is a 5' to 3' exonuclease activity that follows, and that creates then a gap in the DNA. And that gap then needs to be filled in, and the filling in happens in the direction of DNA synthesis, 5' to 3'. So now it actually allows for the opportunity for that region to be resynthesized, and that eliminates the mistake that has been done by DNA polymerase. So this is a not fully characterized mechanism in humans. There are 2 enzymes that are involved, MSH and MLH. They are homologs of the bacterial enzymes, but it is believed that there are potentially other players. It is known that there is a 5' to 3' exonuclease activity that is essential for this pathway, but it all starts with the recognition of the damage by MSH and the cleavage in the double-stranded molecule that is made by MLH. All right, so that's all that you need to know about mismatch repair. Oh no, I forgot something. There is an actual disease association in this pathway as well. So we had mentioned how there is a human syndrome that is associated with defects in global genome nucleotide excision repair, that is seroderma pigmentosum. There's also a human syndrome that is associated with defects in transcription coupled nucleotide excision repair, and that is cocaine syndrome. And there is also a human disease that is associated with defects in this particular pathway, and that is known as the Lynch syndrome. And Lynch syndrome is simply characterized by a very high frequency of cancers, specifically the ones that occur in the digestive tract. And one of those type of cancers that are frequently seen in people affected by defects in this pathway is one known as hereditary non-polyposis colorectal cancer. So this type of cancer is frequently found in people affected by Lynch syndrome. So whenever you have defects in mismatch repair, regions in the body that are undergoing rapid cell division, such as your digestive tract, are prone to develop cancers, because now you have accumulation of mutations. Whenever you undergo DNA replication, there is a higher frequency of mutations, and that predisposes for the development of cancers. And Lynch syndrome is then associated with gastrointestinal and reproductive system cancers, with the main organs affected being the colon, the endometrium, and the stomach, endothelium, and the ovarian regions. So individuals that are affected by Lynch syndrome typically end up developing cancers in one of those regions, epithelial cancers in one of those regions. All right, moving on. The last two mechanisms, we're just going to touch base very briefly. The first one, translation DNA synthesis. There is a whole movie that is specific of translation DNA synthesis already in our YouTube channel, so you can just take a look at that movie. The only important point that I want to make about translation DNA synthesis here is that it is not really a repair mechanism. What translation synthesis is all about is the ability of some DNA polymerases to continue synthesizing DNA in the presence of cyclobutane rings, specifically thymine dimers. So whenever you have thymine dimers, there are specific DNA polymerases that have the ability to recognize that that's a thymine dimer and that the strand that is being formed complementary to that thymine dimer should contain two adenosines. So this enzyme has the ability to put the right nucleotides in the nascent chain that is being synthesized, in spite of the fact that there is a cyclobutane ring in the strand of DNA that is being used as a template. So translation synthesis is all about being able to read over a thymine dimer, being able to synthesize the strand that needs to be synthesized, even though there is a thymine dimer on the strand that is being used as a template. So it ignores and bypasses the damage, but it doesn't fix it. And therefore, it cannot really be considered to be a true repair mechanism. It's just a way to ignore the damage and bypass it for the time being, so that it can be fixed at a later time. Now, these enzymes, these DNA polymerases that have the ability to do that, to bypass those thymine dimers, are nevertheless error prone, which means that even though they know what to put in the nascent strand, even though there is a thymine dimer in the template, they make a lot of errors in their normal activity. And that is because they lack proofreading activity. So they don't have that proofreading activity that is present in our highly processive DNA polymerases, DNA polymerase delta and DNA polymerase epsilon. All right, so that's all that I needed to mention about translation DNA synthesis. And the last mechanism of DNA repair is recombinational repair. And again, this is another topic that I want to touch base, but only very briefly. And the only thing that I want to mention about this is that the type of DNA damage that is repaired by recombinational repair is the type that can have the most damaging consequences for us. And that is the type of damage that triggers double strand breaks. So think of it. The damage that we have talked so far in our DNA is the kind of damage that affects one of the two strands. So for instance, whenever you have cyclobutane rings, it's using one of the two strands. And then the mechanism to fix the damage is you eliminate the strand that contains the damage. And once you eliminate that strand, then you use the strand that is still in good shape you use that as a template to synthesize the region that contained the damage. So basically, the idea behind this is that in every one of the mechanisms that we have talked about so far, one of the strands is not being damaged, so it's still intact. and the damage is present in only in one of the two strands. So you can use the other strand as a template to fix the damage that happened in the complementary strand. But in this particular case, whenever we have double strand breaks in the DNA molecule, the mechanism that will allow you to fix that kind of damage is substantially different, because here, in this particular case, now you don't have an intact strand of DNA to fix it. So you need to invoke a completely different mechanism to be able to repair this. Okay, so important question. What triggers double strand breaks in our DNA? The main cause of double strand breaks in our DNA is by far ionizing radiation. And ionizing radiation is just a type of radiation that happens in a different region of the energy spectrum that we are so familiar with in our everyday activities. So keep this in mind. Electromagnetic waves come in different wavelengths. Electromagnetic waves come in different wavelengths. And depending upon that wavelength, that kind of electromagnetic waves can manifest themselves as visible light or infrared waves or microwaves or radio waves. And all of these are toward the low frequency side of the spectrum. That means the frequency of those waves is lower and lower and lower. And on the other end, you have the high frequency type of electromagnetic waves, and those, the higher the frequency, the more those waves will actually interact with matter, and the more they will interact with our atoms, and the more they will actually have the ability to affect the normal chemical structure of all the components that make us. So on the high frequency end, we have ultraviolet waves, and those we already know what they can actually do. Those can actually trigger the formation of those cyclobutane rings. And then we have X-rays, we have gamma rays, and then we have cosmic rays. Now, X-rays and gamma rays are among the two that we actually do get exposed a lot whenever we undergo certain medical procedures, such as X-rays, and those type of electromagnetic waves have the ability to create double strand breaks in our DNA, and that's why exposure to them can be quite damaging. So what happens whenever we have double strand breaks in our DNA? Well, we need to fix it, and there are different ways to fix it. One of them is by using a mechanism known as non-homologous end joining, or NHJE. Non-homologous end joining simply invokes taking those ends that had been produced and trying to basically just reconnect them together. Now, the issue with this is that whenever you have a break in DNA, there is a very high frequency of having the initial break triggering the activity of exonucleases that will chew up on the free end that has been produced, therefore producing substantial losses in the sequence of the DNA. So those are deletions that are now being introduced into your DNA, and that happens whenever you have free ends. And this is a very important idea. Any kind of DNA break may actually trigger the activity of exonucleases, and that will eliminate substantial regions of sequence at the ends of the breaks. And if you were to simply take the broken pieces and then connect them again, the resulting structure that you will generate by connecting those ends that have broken up is very likely to contain substantial deletions. in the sequence of the DNA that is produced. However, this mechanism actually does happen in our cell. Non-homologous end joining actually fixes a substantial number of double-strand breaks in our DNA. But again, this is a very error-prone DNA repair mechanism. Now, the other alternative to just simply connecting the two ends that had been produced by those double-strand breaks is to use general homologous recombination. And this mechanism of DNA repair is, for the most part, error-free. Meaning, if you invoke homologous recombination as the way to fix those double-strand DNA breaks, the resulting molecule that will be produced will be error-free. All right, so let's just have a very quick review of what is required for general homologous recombination, and this is an essential idea. So whenever you have ionizing radiation hitting your double-strand molecule of DNA, those breaks that are produced in your double-stranded DNA molecule could be fixed either by non-homologous end joining or by homologous recombination. Now, this mechanism is error-prone, and typically it leads to loss of bases at the ends of the DNA molecule, whereas homologous recombination is basically an error-free mechanism of DNA repair. Now, important, I don't know if you guys are familiar with CRISPR-Cas9, but CRISPR-Cas9 actually uses the idea that you can actually target specific regions within our DNA for breakage simply by using the specificity that is associated to this system. So you can actually use a guide molecule so that you tell the CRISPR-Cas9 system where to break into your DNA, and by doing that you can actually inactivate very specific genes, or you can trigger the insertion of new genetic material in very specific locations of your genome. But the mechanism by which the breaks that are typically triggered by CRISPR-Cas9, the mechanism by which those breaks are typically fixed is by non-homologous end joining. All right, so continuing, homologous recombination then is the alternative, and it is fairly error-free. But the requirement for homologous recombination to take place is you need to have an identical molecule of double-stranded DNA to the one that has suffered the break. And that basically invokes the presence of a sister chromatid, the type that are produced after DNA replication. In other words, homologous recombination is only possible after the S phase of the cell cycle, meaning it is only possible during the G2 stage of cell cycle. Homologous recombination is only possible in cells that have already replicated their DNA, cells that are in the G2 stage of the cell cycle, and in fact it can actually take place while cells are entering into mitosis. Now, this kind of DNA damage, double-stranded breaks in the DNA, are among the most damaging types of DNA damage that we have. These breakages are actually associated with DNA duplications, and you can imagine how if you excise a whole segment of DNA, you can then introduce that segment that has been excised from one chromosome into another chromosome, and you may end up with a duplication. Whenever you have those breaks in one chromosome and that segment has been excised, it may actually be fixed, but in the flipped orientation. So you end up with a flipped direction, and that's referred to as an inversion. Or whenever you have those double-strand DNA breakages in your chromosome, the segment that has been broken may simply be lost, and upon DNA repair you may end up connecting segments of the chromosome but without retaining the region that has been excised, so that's a deletion. And sometimes the pieces that are breaking apart end up landing in another chromosome, so they insert into a different chromosome, so those are referred to as insertions. This is important because sometimes insertions are actually responsible for trisomic phenotypes. So chromosome 21, the trisomic form of chromosome 21 sometimes is simply associated to an insertion in which a big segment of chromosome 21 ends up inserted into another chromosome, therefore producing a pseudo trisomic stage, which still leads to the full spectrum of the disease, Mongolism, what we refer to as Mongolism. And that sometimes is due to insertions. And then translocations also take place, in which a segment ends up also translocating together into another chromosome, but the two segments are actually interexchanged from one chromosome to the other. That's what is considered to be a translocation. So all of these type of chromosomal aberrations, chromosomal damage that ends up with aberrant chromosomes are due to this type of chromosomal breakages. So again, whenever you have a break in both strands of the DNA, therefore breaking your chromosome altogether, that kind of damage is the type that can lead to the most damaging type of effects in your genome. All right, moving on. So again, general homologous recombination requires the presence of a sister chromatin. Sorry, I'm tired now. Sister chromatid in your DNA. So it can only happen in cells that have already undergone DNA synthesis. So again, it happens only in cells that are in the G2 stage of the cell cycle. And again, this is just a very brief graph of the cell cycle. You have mitosis, you have G1, S phase when DNA is synthesized, and after S phase, every chromosome now remains associated to its sister chromatid, therefore having two chromosomes that are identical to each other and that remain associated to each other all the way until when the cell enters into mitosis. And again, these chromatids are identical to each other, so any segment here has an identical segment on the other chromosome. So if there is a double strand break in one of these two copies of this chromosome, it can be fixed by using the other copy. And that's the basic behind homologous recombination. And let's go into the process fairly quickly. So here you have a double strand break in the DNA. So both strands are broken. And the next thing that happens once you have that kind of DNA damage is you have exonuclease activities that are going to show up into the broken molecule of DNA. And the type of exonuclease activity that is depicted is a 5' to 3' exonuclease. Now that creates regions of single-stranded DNA, and those sequences contain identical regions in the other sister chromatid that is present in very close proximity to a broken chromosome, because again, this is in G2 stage of the cell cycle, so you have two identical copies of that particular chromosome in very close proximity to each other. So that single-stranded segment of DNA can then invade the other strand, and the process of invasion is actually driven by the engagement of another protein, one protein that has the ability to bind to the single-stranded segment of DNA that has been generated and drive that single-stranded segment to invade the double-stranded DNA molecule that it is homolog to. All right, and the process of strand invasion generates this kind of a structure that we refer to as holiday junctions, structures that look like this. So you basically have one DNA molecule invading another DNA molecule. And holiday junctions, two of them are actually generated during the process of strand invasion during homologous recombination. Okay, so continuing. Once you have undergone strand invasion, now what you have is basically a free 3' hydroxyl group that acts as a primer, therefore allowing for DNA synthesis to take place. The same is happening here. You have a 3' end that can lead to DNA synthesis so that you fill in the gap, and that is what is represented here. So after DNA has been synthesized, now you have fill in the gap. You're using the intact chromosome that was not broken as your template. And that is why homologous recombination is error-free, because you are still using a template to drive DNA synthesis so that there will be no mistakes in the process of repairing the double-stranded molecule that was broken. Now, once you have filled in the gaps by DNA repair mechanisms, there are different ways to resolve the holiday junctions that are being produced. And depending upon how those holiday junctions are resolved, you may end up with hybrid DNA molecules or fully recombinant DNA molecules. But we are not going to go into the details of that. The basic idea is that at the end of this process, you end up repairing the double-stranded break that took place. Now, the main enzymatic player that you need to be aware of here in this process is the protein that drives the invasion of the double-stranded DNA molecule by the single-stranded segment that has been produced, and that protein is RAD51. That's the protein in humans. The bacterial homologue for RAD51 is REC-A. So REC-A is the one that is responsible for homologous recombination in bacteria. All right, now these events, all of these processes are substantially more complicated and substantially more involved in terms of their actual molecular biology. But for this introductory class in molecular cell biology, this is all that you need to know related to homologous recombination. So with that in mind, then I just want to again give you this final outlook to what we have actually talked about. We have talked about the different mechanisms of DNA repair. We talk about direct reversal as being the most effective, most rapid, most phenomenal way of repairing DNA damage. Then we talk about excision repair, and we talk about how this is the most flexible, the one that can repair the vast majority of damage that happens in our cell, excision repair, specifically nucleotide excision repair. And then we talk about translation DNA synthesis, which is not truly a DNA repair mechanism. And then we talk about recombinational repair, which is involved in fixing double-stranded DNA breaks. So with that in mind, I want to conclude this. It was substantially longer than I expected. Sorry for that. And this is not going to be edited. It's going to go basically the way that it is being recorded into our web page. Sorry about that. It's about an hour long. I hope that you guys won't die listening to this video. It should have been half an hour long. It ended up being an hour and 11 minutes long. Sorry for that, guys. Okay, well, this is another movie.